for those who don't know me, I'm work for the U.S. Department of Agriculture. I'm located on Cornell's campus, and I am most of the time a maize geneticist, but it's been truly my honor over the last nearly now a decade to be working with the folks in cassava. And over that time, it's really working with both these species has really reshaped a lot of my thinking on lots of things. And it also just been an incredible honor to work with this community. So I want to talk about how our group and the collaborations across both maize and cassava, we're starting to think about how do we move genomic selection to a process that's based on molecular activity. And the work that I'll be telling you about has been supported by USDA, NSF, the Gates Foundation, and as you see, also some work by USAID and NIFA. So, the cycle we've been working in for the last century is pretty much this. Selection, crossing, evaluation takes 5 to 10 years. And what NextGen Cassava was focused on was how can we shift it to something more like this, where we're able to do genomic prediction under much more rapidly for whatever traits that are needed in order to make those decisions on selection and crossing more rapidly. And I think, over and over again, people have been able to show that genome-wide production works well, but it has limitations. So when you've got a good training data set, big population, you can make accurate predictions. If you take those models and you try to predict across populations, those prediction accuracies can drop dramatically. And if you are working in a specialty species with a small sample size, just from that, you can have relatively modest prediction accuracies. And essentially, we're not leveraging any information from one species to the next. And so the question we've been asking over the last few years is how can we start adding in our mechanistic knowledge of molecular biology and molecular activity to improve these predictions? It's not going to replace these. It's, how can we take that standard genome-wide prediction model and give them a boost, especially in the context of between panels, when we have small training data sets and so on. And sometimes you hear that, oh, genomic selection falters because it's not mechanistic, and that's not true. Genomic selection is mechanistic in terms of it mechanistically models inheritance. But what it isn't doing is not mechanistically modeling molecular activity. And so what we want to really see is how do we put the two of these together? And some of this also, some of the benefits we're looking to get have to do with dimensionality reduction and adding functional constraint to these models. If you just look across the genome in cassava, you'll see 20-some million genetic variants. But if you break it down in terms of the number of proteins that are active and how the range of different mRNA or protein levels, what those levels are active, they're going to be much narrower. And so we think by essentially shifting down to these activity models, we may be able to achieve something like 175-fold reduction in the dimensionality of the number of predictors that essentially are going in to predict whole plant phenotype, and also functionally constrain those. And so potential benefits of molecular activity genome-wide prediction are increases in accuracy. It could be more transferable across populations and maybe even some between species, inference across rare alleles, and it would reduce computational load. And so we have some examples already that it does work in maize. Andrew Geary in the group led an experiment where she was imputing mRNA expression levels of haplotypes to improve genome-wide prediction. She saw that the cis component, the key component that we wanted to impute, was only about 1/3 of what was out there, but it was very transferable across tissues. And then it gave her consistent improvements in genome-wide prediction over standard haplotype prediction. Guillaume Ramstein in the group was looking more at the protein side of things and was trying to improve genome-wide prediction of maize hybrid yield. Actually, a very hard problem is to improve those yield predictions that we've struggled on for a number of years. But what he was able to see was by adding information in on protein structure and the individual disruption of individual amino acids and training that against evolutionary constraint with a machine learning model, he was able to consistently get substantial improvements in hybrid yield prediction. And the interesting thing was the number of relevant variants for making those improvements was very modest. On a per population basis, you're talking on the order of hundreds of variants, even though there was millions out there. And on a per gene basis, you're talking the order of 15 to 100 variants were really kind of contributing to these accuracy in these models. So the question is, how do we build that type of biological knowledge into this great system that Lucas Mueller has been leading with Cassava Base and Breed Base, and we've been leveraging in our breeding insight efforts? And so this messy diagram right here is essentially one blueprint for kind of doing this. And I'm going to break it down where this diagram is coming from. So I also want to only focus on genotypes because, of course, there's lots of very important tools for tracking other aspects of what people are doing as breeders. But the current system for genotypes is we take mid-density genotypes, combine them in breed-based, and then with the Cassava-based interface, breeders and geneticists are working with that data set. We'd like to start adding a haplotype graph to it, and I'll describe where we are with that. The haplotype graph we've been using is the practical haplotype graph, developed by Peter Bradbury, Zach Miller, Lynn Johnson, and Terry Cass-Stevens. And what the PHG really is, it's a representation of a pangenome. It's a database, and it's really then a computational framework and an imputation tool. And over the last few years, they've been able to build that system and then also connect it up to BRAPI over the last few months. And so what it allows us to do is to essentially estimate haplotypes across the genome and see which ones are shared between various individuals. And then we're able to take skim sequence or other types of genotyping approaches, map them across those haplotypes, and for imputation, determine what's the best predicted genotype. And so Evan Long has been applying this for Cassava. And the real problem in any haplotype graph is to really get those phased haplotypes first. And he was using regions of identity by descent, or IBD, identified based on runs of homozygosity, to identify those and populate the practical haplotype graph. And he's got a paper on that just came out. And the key thing to note is that essentially the Cassava PHG imputation accuracy was better than the standard of the art Beagle at most low coverage and across the various data sets we had. It doesn't, but of course, you only have a subsample of all the total haplotypes out there, and at very high levels of coverage, it wasn't doing better for those rarer haplotypes. You know, he also has carried out simulation to suggest that if you do have high accuracy and a complete knowledge of those phase haplotypes, then the practical haplotype graph would, on average, do better all the time. And so really the question is, how do we make sure we have a good capturing of all those haplotypes that are available out there? And so Bethany Econopoulos in the group has been going through with some of the nanopore data that was collected actually a little while ago in the group, but wasn't as useful as we might have liked it to be, because essentially the base callers at the time and the technology for interpreting that data wasn't as good as it is recently. And there's been some really important improvements there. We're finding that essentially going back to our old data, reanalyzing it with a new base caller, we're getting substantial improvement in the assembly of genomes. And so I think I'm very optimistic that over the next several months, we're going to have a series of nanopore assemblies of very high quality that can be combined with other efforts that other groups are doing to essentially pull together the haplotype graph. And I ask everybody across this group, anything you want to kind of get into this haplotype graph, let's work on working together and making that a reality. So we can have a haplotype graph that essentially we then now need to start working with. And you know, we've spent a lot of effort recently on making these tools more interpretable. Brennan and Monnier in the group has essentially created a tool to work with the PHG called RPHG, and so it works in your regular R, produces table reports, visualizations, HTML reports, and so on. He's also worked with Terry Cass-Stevens to connect up our software package R Tassel to R, and so that you can use all the tools we have on doing natural diversity analysis, LD, and GWAS and so on with an R interface. And then both of them have been made BRAPI enabled. And so essentially, you can now start connecting the two of them together. The last element we really need to pull together is the fact that these databases are big. And we're talking about very big data sets. And through a Feed the Future project, ILSI, Francisco and Bethany have been working on how do we get some of these tools up into the cloud and analyzable from there. And so they've been getting Jupyter Hub and RStudio Server enabled so that you can use those tools that we've just been talking about, but essentially be just logging in from wherever you are in the world. And I think the last element is how do we get reasonable speed internet to all around the world? And I think satellite internet over the next, as that rolls out in the next couple of years, will essentially allow people wherever they are to have incredible power to do analysis. So that's how we're working along that top element of it. And so let's finish up here by how do we add in molecular activity models that kind of motivated this whole process. And essentially, how do you get in evolution? How do you get in protein structure? How do you get the knowledge of expression in? And so when we're talking about getting the knowledge of evolution in, and that was really key for the calibration we were showing about protein structure and evolutionary constraint that I showed you, this wins that Guillaume Ramside had in maize. Evan Long is following along that route by essentially sequencing a wide range of different U4BAC, some with long reads, some with short reads, and essentially starting to pull those together so that he can do a similar type of analysis where he can look at the impact of various amino acid changes on protein structure and disruption. And so I think that will give us a powerful catalog of deleterious disruptive amino acid changes in various proteins.
Hey, Ed, I think we have about 2.5 minutes left.
Okay, thank you. And so the other area is, I think, there's incredible tools on coming out of other communities such as AlphaFold 2 for predicting structure directly from these. And then finally, I think there's an open question in our community is how do we get the best measure of mRNA expression that we can impute across all of our samples? And I think we can leave that for a later discussion among our community. So I think essentially the basic tools to achieve this are coming. And I think the key thing is we can pretty much hide a lot, except for a few people, hide the details of this. And essentially people can be working in what is their comfortable way as a breeder or a geneticist with a lot of this, but have incredibly powerful tools to make better predictions. And I'll end there and just mention that this really wonderful work has been led by Bethany, Evan, Brandon, and Cinta, and then the PhD work by Peter Bradbury, Zach Miller, Lynn Johnson, and Terry Cass Stevens.